Hello, everyone. We are ready to start our third installment of the Journal Club at the Lab School at IVF Academy USA. Today's topic is first evidence of microplastics in human ovarian follicular fluid and emerging threat to female fertility. Before we get started, I just want to talk about a few housekeeping things. Please, this is an audience engagement is requested, so please unmute yourself, turn on your camera. Feel free to update your name to your first name and your last name as the city you are in, so we know where you are. Submit questions anytime. Feel free to interrupt, come offline. Additionally, you will all be a link to the recording of this event. I am now going to turn it over to Dr. Anderson, who is going to lead today's discussion.
Great, thank you, Lygia. Glad everyone's here. Got a really good turnout today, it looks like. Want to just kind of open it up to, this was a paper recommended by Brittany Garza. Should be here any moment now. And but I thought it was a pretty good paper talking about the microplastics and nanoplastics in follicular fluid. There's been some discussion on this context of the plastics being in seminal fluids in the past, and just thought I'd open it up because, I mean, I have my own opinions, but I'd like to get an idea of what everybody else thinks. So you can see around the room here, we have our current students here at the IVF Academy, and we do this journal club every other week. And it's a way to just kind of bring out the papers, talk about more things in the literature, and just to be more aware. So I'll leave it up to open up the students. If you have any ideas, please share your comments.
Questions or comments?
Any comment, question, anything?
I have a question I wrote down about this article. And when I look at articles, I read everything as in the question in. What factors might explain why no link was found between microplastic levels and outcomes like fertilization, miscarriage, or live birth in the study?
That's a really great question.
I thought
one of the things I looked at when I saw in here, it does talk about, if you look at here, the FERT rates were really good. Their pregnancy rates, they had a 47% pregnancy rate, but they had which isn't bad. And it doesn't really, I think the average age was 35.9, just kind of average for a lot of fertility centers. They had a really high miscarriage rate. 23% is really high miscarriage rate. So was that due to microplastics? There could have been a lot of other variables, like what is this center's normal miscarriage rate? So as a miscarriage rate, there's ways to define pregnancy. There's positive beta, which is what they're saying here, 47% positive betas. So let's say if we have a 63% positive beta like in our fertility center upstairs, we probably lose around 10 to 15% of those, which is about average. It's a little less if you're doing genetic testing, because to genetic testing, they didn't mention that here. So 20% is a little bit high. So it could be due to other variables too, like progesterone. So you're right. It didn't really seem to affect the other parameters. The FERT rates were good. I thought it was kind of odd that their blast rates, I don't know if this is like a percentage of blastocysts, but 91% blast rate. That's way better than my blast rate. We have about what is our blast rate? Probably around 60% blast rate. And 40, anywhere between 35 to 40% usable blast rate. So 91%, that's pretty high. So that's a good question though. And so, it does seem to affect things. I talked about hormone stuff in there, but it doesn't seem to affect the actual outcomes that much.
Considering there is no effect, what if we assume that there would be an effect? If the microplastics were in a high concentration, it said that the granulation cells were affected. So that be considered a reason why many more women are susceptible to PCOS, because that those granulosa cells, a lot of those androgens aren't being converted, so there's the increased testosterone.
Well, does anybody else have any comments on that? The granulosa cells, like have an effect on PCOS? PCOS is just a reason for infertility. It's a diagnosis. PCOS really, what PCOS means is that the polycystic ovarian syndrome, like, and there could be various degrees of PCOS. And so, as an example, using my personal life as an example, my wife is slightly PCOS. She had a period about every 40 to 50 days. She wasn't the textbook 28-day. So what that means is that she has fewer periods per year, fewer opportunities to get pregnant. But once she did have an ovulation, she was able to get pregnant. So we didn't have to do fertility treatment. Just recognizing that. So just, it's not so much that it was maybe causing PCOS. PCOS just happens naturally.
So PCOS or increased testosterone isn't what would cause PCOS, it's more as a secondary effect of PCOS?
Yeah.
Oh, okay.
I mean, it did talk about, possibly affecting the granulosa cells, and those are responsible for secreting estrogen and progesterone. Like, it would be interesting to be, but I just don't know how you would pinpoint the fact that if those microplastics are being exposed in the granulosa cell, are they hindering the production of estrogen and progesterone? And there's so many other things that could be also indicative as to why they may not get the response that they were looking. I did think it was really neat that they use like glass products to try and rule out any of those plastics. We get the best of the best plastics, MEA tested, and we still kind of see those plastic shards in follicular fluid. And I never have once said, oh, that came from the follicle. I've always assumed it froze from the dish. And they are bigger than the 5 microns that they are referring to, with how large the microplastics that they did find in the follicular fluid. So it's kind of hard to say, like, what specifically is the reason why, but they did say that, there could be some correlation there.
I think about 5 microns is pretty big. I mean, I was trying to see what's the largest thing that could cross the cell membrane. And, who do you ask? ChatGPT.
Yeah.
So I asked ChatGPT, like, what's the largest? It said 500 Daltons. I don't know how big that is, but it sounds a lot smaller than 5 microns. Actually, I did say, like, what is it? That's, Daltons is a mass, not so much a size. But yeah, 5 microns, that's pretty big. You think about a zona pellucida is 15 microns, so it's probably 1/3 of a zona pellucida on average, which you would think you could see under the microscope. Any other comments? Students?
I do wonder how they would be able to, I don't know if they would be able to, but if they were to see how much like a pregnant woman has in her system compared to those who aren't pregnant, see if like, if there's a comparison there because they're already pregnant.
Yeah, right. Yeah. I think that's kind of a great follow-up study. Like in this context, I think you mentioned earlier when we were talking, culturally different parts of the world, right? So where maybe this was done in Italy, but I thought what's interesting is that they had a lot of losses. They started off with 80 patients, 37 patients were excluded. Samples had blood traces.
Yeah.
So why were they excluded? Because I don't understand that. 25 patients just didn't respond back, right? That happens. So it got down to where there were 18 patients. And when I first read the study, I didn't actually catch that they were collecting in glass. I'm like, oh, that's where it's coming from. It's coming from the plastics, but they did collect in glass. But if you're collecting through a tube, the needle has a plastic tube on it too. So could it be coming from those? And Danielle pointed out that there were 3 actual blank samples ran. So they did run a blank to identify if there were, so they did have like a control sample in there, right? So, so is this truly coming from the follicular fluid or is it from, outside? But it does sound like there is some in the follicular fluid. And it was, peer reviewed and, thought to be published. So, accepted that the evidence does show that it is in the follicular fluid and publishable.
Hi. I think that's something that I found super interesting was that like they found some patients that they didn't have any microplastics in their follicular fluid. Like they say it's like 0, which I think is a little bit extreme because like if they're all having like kind of the same lifestyles and diets and they all say like they all like consume the microplastics, like well, they are eating from like plastic bags and like water bottles, and that's like our main like understanding that's where the microplastics are coming from. How come we have 3 patients or 4 patients that they don't have anything in their system, like literally 0, and then we jump from 0 to 442? I thought that was a little bit like misleading, and they never come back to this after in the discussion, like how come there's some patients that have 0 microplastics?
Yeah,
That's a great comment. Thank you, Samantha. I thought the same thing. It's like there's 4 that had none, but I think one of the requirements was to get in the study that you had to be drinking from plastic bottles for at least, was it 3 years? 3 years. So had to be drinking from plastic bottles and utilizing plastic materials for at least 3 years.
And it's not even like it's a low number, it's just 0,
Yeah, I think you would have something detectable in there.
at least, yeah, a little bit.
I would have liked to have seen more, like 18 doesn't seem like an awful lot, like if you had 100 or 1,000, what would you find? They did have a lot of, go ahead, Doug, what'd you say?
I said that is interesting about this paper that they constantly acknowledge that their n value is pretty small. Like they'll make a point and say, oh, well, we need more research because we only used 18, a sample size of 18. So it's like, I'd like to see them like redo this, or even maybe like, not to say that this shouldn't be published, but like, why not redo this with more people and then kind of finalize, your conclusion rather than kind of stir up a bunch of mess by kind of concluding that there's microplastics and it's because of what you saw in 18 people.
Yeah. We were talking about that earlier in the training lab where, in a lot of science, in a lot of publications, a lot of people will, they take one reading of something and then they make a lot of medical changes based on, it's like, what's evidence-based medicine to make this decision to do that? And I'm not saying that there isn't evidence here, but, the idea is that more studies need to be done rather than, making that this is the clear outcome of that. And I think as scientists, and that's kind of what I want to encourage the students or anybody in the audience, is the idea is you want to kind of always be questioning things, what, just because it says it, and that the idea with science is that sometimes science is wrong. And even when you're a scientist, if what you said 10 years ago is different today, to, redo that work and say, hey, what I said 10 years ago or 20 years ago is wrong. Here's the updated data. A good example of that is Good to Great. I actually recommended the book Good to Great to somebody. And it's a great book in the sense of leadership. If you read my newsletters online, I've been talking a lot about leadership lately. And somebody replied back to my message, like every single one of those businesses in the original book have gone out of business. But there's an updated version of Good to Great, that the principles of that book still stand true. And so the idea is that new science, new data, new literature actually is really good. And I think I said this last time that like we talked about DNA fragmentation, we talked about reactive oxygen species. I think some of that original data could be redone and would make great master's and PhD projects for anybody that's in the audience.
I mean, if it's 14 out of 18 in Italy, I would be very curious to see what it is in the States.
I know.
I mean,
While you drink out of your plastic water bottle.
yeah, cheers.
You know, I drank my protein shake out of a plastic container, 20 minutes ago.
Yeah. I mean, let's not even talk about the straws, Most of
Right.
them are plastic.
In the styrofoam cup. Styrofoam is nothing more than aerated plastic.
Well, yeah, this opened the questions of what can we do in the lab? How can we minimize plastic in the lab?
Yeah,
I mean, it does make you aware of what are you using in the lab.
yeah.
Talk about that time when we didn't have plastic back in the day, Tony. How was it like having nothing but glass?
Why, just because I have a lot of gray hair? Think I used to use glass?
I read about it.
You think me and Bob were like this?
Yeah, how was the day when there was no plastic?
By the time I get into human IVF, they were still plastics. I even got in late enough. I got into human IVF and did bovine in 1991. I got into humans around 1992. And they used to actually, they call it test tube babies. They would actually take a tube of media in the bottom, put your egg and sperm in there and let it fertilize that way. And you had to actually go in and pull the egg out of that, put it into fresh media the next day. By the time I got into human IVF, we were actually using 4-well milk dishes. But if you think about it, that's a great example, though. I was showing the lab team in the lab and the students in the lab. I share this with everybody in the context of there's different types of plastics, right? And so a lot of people, back during COVID, we couldn't get the typical Falcon 2095. That's made out of polystyrene, right? Polystyrene is a really hard plastic. I meant to bring it in here. Like if I were to take a hammer and hit it, it would shatter into a bunch of pieces. But I could take the tubes that I have in the training lab, because it's for training, that they're made out of polypropylene, much more flexible. I could practically bend it in half and it won't break. So you could use that as an example. That the plastic there is a soft plastic. It's a very thin plastic that your water's in. It's it's very flexible. But if you look at like a Nalgene water bottle, it's very hard plastic. Nalgene is supposed to be really good plastics, and the medias that we order come in a Nalgene plastic. So it's a really good hard plastic that doesn't leach as much into it. But I will say that some of our medias, if it sits in those tubes long enough, are probably leaching plastics into them, right? So if you aliquot your medias and you aliquot too much, you might be leaching those plastics into the media and growing that into that. So use aliquot what you're going to need, and then maybe like on a weekly basis, so you're not pre-aliquoting. I have an example of this where I was, a lot of us use the airtight syringes, the plastic syringes. You can take those green plunger syringes, you can bend it in half, it doesn't break. That's polypropylene. It's probably the least toxic syringe. You could add media to it and use that for transfer. Within, for under 10 minutes, it's not going to kill mouse embryos or sperm. Leave it in there for an hour, though, and that media out of that syringe will actually kill mouse embryos. And when I used to work with Klaus, he said you could grow a mouse embryo in Budweiser. So I never tried it, but maybe that'll be our next project.
Because Budweiser comes out of a can.
Yeah, exactly. As long as the pH is right.
Yeah.
But no, it's a great point. So we do use very hard plastics that are glass-like. And I think the harder the plastic, the better. And so not all plastic is created equal. When I used to do bovine work, we used to weigh our glycerol because we used to make our freezing medias, and we would weigh it, and we would weigh it every week. The reason why is because if you left that glycerol in there, and if you've ever seen glycerol, you could, it's like PVP, it's thick like PVP. You could turn it upside down and just like barely move. but by the end of the week, it would turn into like a water-like substance. So it was actually changing its chemical property by just sitting in the plastic tube. Like the plastic polystyrene tube that we use in our everyday lab. So something to think about. So it's, it could be being found in the plastics, but like you're saying, there's a good chance that we could be introducing these things in the IVF lab. So good way to improve things. Has anybody in the audience got any comments that they'd like to add? See if I recognize any names, I can call you out. Emily Wright, you're a past student. You read the paper?
Sorry, I had to unmute myself. No, I didn't. I actually just hopped on today to listen to what you guys were talking about. I'm sorry.
No, that's okay. I'm sorry to call you out like that, but see if
Brandy.
is Brandy on there? Brandy, did you read the paper? Oh, Brandy Patino, I see you up there.
No, I actually just hopped on as well. I saw it this morning.
Great. Oh, that's good. I'm glad everybody's here. What was another kind of actually, Danielle, you mentioned aluminum.
Oh. So I saw on page 5, there's this graph. And it didn't really talk too much about it within the paper. But there was like this big spike of, I'm assuming aluminum that they measured, which I thought was interesting. But I also saw within their process that they use aluminum for they preserved a glass box and protected by aluminum foil. So I don't know if maybe there was some type of contamination or if that was what was really within the sample.
I have a comment on that actually.
Ooh.
That's not a graph. Actually, it's a chemical composition reading. So like a HNMR reading. So you have a sample of something and you put it on like a almost looks like a spectrometer, and it'll tell you. So based on these peaks, it'll tell you the chemical composition. So it's not really saying there's more or less aluminum. It's just saying that spike at that wavelength, I guess, is aluminum, and it has it labeled as aluminum.
Okay.
But why is aluminum in there? Where is that coming from?
That's a good question. I don't know.
And it doesn't really talk about it in the paper that we could find. And so, yeah, it's just an interesting, just kind of a side graph. Couldn't figure out what they were trying to say with that. It talked a lot about how they were testing hormones on things as well, in the materials and methods. It doesn't really break down much of their hormone evaluations on anything. So I didn't see where that was broken down in the paper. But anybody else read the paper and has some comments on that?
So I asked ChatGPT, and so the aluminum is in the graph comes from the aluminum stub used to mount the samples from the SEM imaging. That's how they got the results.
Okay.
So it's not part of the microplastic.
It's used for the mounting. Okay. And the processing of it. See, ChatGPT. Those areas.
Okay.
So, and recognize this was done in Italy. I thank you. Like we mentioned here a bit ago, saying that this might have, how would this look in other countries? It does actually talk a lot about, it actually talks a lot in here about feeding mice the microplastics and actually looking at their ovarian response to that and testicular responses to that. Didn't really say how much they fed them. It's kind of like, you know, if you feed a mouse, you know, fake sugars, low calorie sugars that, you know, it says right on the packet may cause cancer, but we're not eating spoonfuls of it. The FDA didn't think it was bad enough that we could ingest it ourselves. I suppose that, you know, is it a ratio of what would a normal human would have, you know, based on kilograms? Don't know. But, you know, I guess if you get enough microplastics in you, you might actually have some hormonal responses to it. I think one of you guys, what's that?
In the animal studies, it showed that it had lower estradiol testosterone levels. So it could
Right.
affect your hormones, and also it showed irregular cycles, reduced ovarian mass, and
Maybe that's why all of 25-year-old or 30-year-olds men are going on testosterone these days. Too much microplastics.
could be the answer.
It could be why.
Interestingly enough, it says that the polystyrene microplastics led to greater accumulation and oxidative stress in ovaries compared to testis. So like it affects female reproductive systems more than it does male.
Oh, yeah, makes sense.
I think someone pointed out, one of the students earlier that they noted was a huge decrease in sperm concentrations over the years since the 70s. Is that right? What's that?
62%.
52%? 62%? Yeah. Anybody have any thoughts on that? Like since the 70s till now, like 62% drop in sperm counts? Any?
Asbestos.
Before I give my 2 cents on it, so those things.
Might have an idea, but
Yeah, lay it on me.
And sperm counts.
I think it has to do a lot with epigenetics. I just went through a lesson, and it shows about how the diet, when they did animal studies on mice, they found out that if the mice had a high sugar diet, the next generation had it affected the next generation also. So it's transgenerational. So it has to do a lot with the epigenetics of before generations and then those getting affecting
So
fertility.
what I eat today is affecting my children, and they eat will affect their children.
Diet, smoking,
Yeah.
yeah.
That's a really, I actually didn't know, that's a really great point. My thoughts were that, you know, when the WHO came out back in the 80s, they didn't have a lot of data to go from. So they just kind of like, well, here's the baseline. And then 2010 version, they lowered that baseline to instead of 20 million, 15 million.
Same thing with morphology.
And morphology and all of them.
So are you saying that the sperm count hasn't actually gone down? It's just that the parameters have
I
changed?
think we just have more data to go from today. That's just my theory, but that's and you know, if you look at that in the sense of sugars as carbohydrates today, right? You know, they call it the SAD diet, standard American diet is basically a carbohydrate laden diet. And so
Well, yeah, because
we're affecting that. We've been affecting that probably since the 70s when foods like that became more available.
they found out that a lot of infertility diagnosis, it's when they look at it, a lot of the imprint is from imprinting codes. So
So can you reverse it?
design your baby.
So are we not seeing that trend in other cultures where they don't eat as much sugar?
Well, I don't know. That's just it. That's the question, right? So
Yeah, I think Americans would be 16, 17 out of 18. So like, it would be 18 out of 18.
yeah.
I do think it's really interesting that it all like kind of ties back to reactive oxygen species.
Good point.
Like
Mention that.
paper after paper, we're always coming back to ROS and we're always coming back to DNA fragmentation to some extent. And this paper is no different. Here we are again, and it still can lead to the apoptosis of granulosa cells, like you were saying. and ultimately endometriosis, which could be another ovulatory factor of infertility. So I think, I mean, it's crazy, like more ROS, more apoptosis, less babies.
Yeah, people are worried about the reactive oxygen species in sperm, but this is actually, they showed that it's actually found in the eggs as well.
I have a comment as well on that. Like, because they're talking also about like the plasticine, which is like this huge boom in like the plastic use. And I just searched like when is that like considered to like start from? Like, what's the starting point? And it says like 1950. And so we're looking at the things with the sperm count decreasing, and they're counting from 1973. So I'm wondering if it's like the parents consuming like also more plastic since 1950, and then like the progeny like starting to have all these problems with like sperm count, and also like if they're saying that it affects more the female mice than the male mice, like that can also lead to something in humans. Like that's my thoughts on that.
It's a great point.
Yeah.
Yeah, absolutely. If you think about when I was a kid, we used to, when I wanted to make some money, I used to go around digging people's trash, digging out bottles and taking them to recycle them, right? They'd pay me for the bottles and cans, right? There was no such thing as plastic bottles back in those days. And so, whereas today, everything's in a plastic bottle, but you can't even find anything in a glass bottle a lot of times anymore.
Yeah, was it milk delivered in a glass
It
bottle?
was, yeah. And the milkman dropped you off glass, picked up the glass. Great point.
The only thing glass in the bottle now is like liquor.
Some of it.
Let's learn from the liquor business.
Yeah.
Yeah, or the aluminum.
They have microplastics in.
Yeah, well, I will tell you that even in my own home, I brought my lunch in my plastic bowl today, but I think we talked about, there's a great Netflix show where they discovered Teflon, right? And they started making all these pans with Teflon. It's the greatest invention since sliced bread. And then people found out that it caused cancer, but you couldn't get the company to admit that it was causing cancer until somebody finally did actually prove it. So we've been exposing ourselves to this, and that was back in the 70s, I think. So might be a lot of truth in the sense to that we've been causing a lot of these problems for their carbohydrate diet with their plastic and consumptions.
Yeah, it's just one of those things that even if you have the best diet or the worst diet, the common denominator is what it's sold in, which is if it's a salad bag, it's in a plastic bag, and if it's chicken nuggets, it probably came from a plastic bag too.
Yeah.
So it's the exposure that's kind of the same.
Yeah.
It makes me think of the plastic chicken from Costco.
Yeah, I was just thinking the same thing. I had one of those yesterday. No, it's there's not a right answer, but is it reversible? I think nobody's really said anything today, but like there's we talk about heavy metals in our waters and things like that too. And so those have to have a very similar impact, I would think. Mercury's in the water, aluminums. I remember when I lived in North Carolina, or when I lived in New Jersey, they would say you would get your fishing license, and in the book, your fishing rules would say don't eat more than 3 fish from this body of water per year, especially if you're in your reproductive years. So it specifically said in your reproductive years. So, and that was like 1993. I remember reading that. And I came from Wyoming where we ate fish a lot. We went fishing, we ate a lot of fish. Never once had I heard that, like, don't eat too many of these fish because of the heavy metals. So that would be a really great paper. I'm not aware of a paper like that, but if someone is, please share it with us and we can discuss that.
Well, Ironically, I got a message right now. It says research from the University of New Mexico shows that plastic buildup in the brain has increased 50% over the past 8 years. The human brain may contain enough microplastics to make a spoon. Yeah,
Each human brain?
yep. So if you can't beat them, join them. Cheers.
So you can consume more knowledge. That's
Mhm.
See more knowledge. No, that's good. I'll throw it back out to the audience again. If you have any comments or anything you'd like to share? Quiet group this week. Okay. Well, I'm gonna wrap it up here and turn it back over to Lygia. Have another paper ready for you guys to read, so come back again in a couple weeks.
Thank you, Tony. So, when the recording for this session comes to you by email, in that email is a link to register for our next journal club. And remind me of the topic again, culture of viable human blastocysts in defined sequential serum-free media.
Yeah, we're gonna talk about an old paper that David Gardner wrote, talking about sequential medias. And so that'll be kind of right on track with the students, taking it back a little bit of old school and looking at culture medias at the sequential medias. His papers are really good.
And before we jump off, I'm gonna go ahead and ask the audience to just, if you have a topic that you would like to see us cover, just throw it in the chat. Additionally, we have upcoming courses here at IVF Academy, hands-on training in the lab. We've got ICSI biopsy vitrification, a week on embryology and a week on andrology coming right up. Please save 25% when you register. Contact me at admissions@ivfacademyusa.com to learn more about our courses. Looking forward to seeing what's in the chat that comes up, and thank you so much.
Thanks, everybody. See you next time.
Thank you.
Zero-free sequential. Yay.